Hello, welcome back. Here we are in our last mini lesson for chapter 2, basic chemistry chapter. We're going to focus on the last few pages of the chapter talking about different types of, they're just calling it biochemistry. It is the chemistry that's more seen a lot more in living systems, not just general chemistry that you might see in a chemistry class. So in biochemistry, we're just going to cover some major topics and wrap up with the four major macromolecules. Remember, we talked about being composed primarily of hydrogen, oxygen, nitrogen, and carbon, which we'll see a lot in the molecular formulas when we take a look at some of those. So the first thing we're going to talk about is water, and we just kind of got done talking about water in those hydrogen bonds. So water has a lot of unique characteristics because of that ability to make hydrogen bonds, to be polar in nature, to attract to each other. So just showing some representations of some of these characteristics. So the first one, I show an ocean. And the reason why I show an ocean is because water has a high capacity for holding heat. So what that means is, let me write that down. It can take in a lot of heat energy and not raise its temperature very quickly. So since we have the ocean here, and I'm teaching oceanography this summer, we talk about this on the, think about the last time you've gone to a beach, right? It's gonna be a nice sunny day, you're on your beach blanket, you're on your umbrella, and you're like, I'm gonna go to the ocean, take your shoes off, and you start walking on the sand. And it's getting really hot, and you're like, you gotta run, tiptoe to the water. And you get to the water, you're like, it's so cool. Why is it that sand and the water, both receiving the same amount of sunlight, one is really, really hot and one is cold or not so hot? Depends on which ocean you're in, I guess. The reason is the ability of the heat capacity. Sand, which is mainly granite, kind of a rock mineral, it takes heat and it changes its temperature really, really fast. So it has a low heat capacity. It cannot hold on to a lot of heat. Because if you're to go to the beach at night, after the sun's gone down, what's the temperature of the sand? Well, it's pretty cold because it lost all of that heat. It cannot hold on to it. Think of the water as like a heat battery. It can charge up on a lot of this heat and not really change its overall temperature. And if you go to the beach at night, the water is about the same. It doesn't really cool down significantly, because water can take a lot of heat and not change its temperature very much, and it can release a lot of that heat and not change its temperature very much. So for how does that apply to the human body? Well, we are mostly water, 50 to 60% water in all of our cells and our blood and our tissue fluid. So we can hold on to that heat, and that helps to maintain our internal body temperature. And it takes a lot of heat to change, to warm us up. And we have to lose a lot of heat to cool us down to any significant degree. And that's all because of those hydrogen bonds. It allows the water to take up a lot of heat. This picture showing the brain is water is buoyant. It has a very low density compared to other things. And so, no, let me reverse that. It's got a higher density compared to similar sized molecules. And so things like your brain and cells floating in your blood, they can float and be buoyant in that, and that helps to protect one of your most important organs, which is your brain. And then lastly, on the bottom here, I'm showing these little Mickey Mouse water molecules with their charged sides, the negative oxygen and the positive hydrogen, coming to salt, right? Sodium chloride. We made that in our ionic bond in the last video, with a positively charged sodium ion and the negatively charged chlorine. That's salt. Think of what happened the last time you made salt water, like you're cooking pasta or something. It dissolves. It just kind of disappears. We didn't really make the salt disappear, because if you're to taste the water, it's really salty. But what we did was water can come in, the positive sides of the hydrogen surround the negative chlorine, the negative sides of the oxygen surround the sodium positively charged sodium ion, and it pulls it apart. So water is a great, what we call solvent. It dissolves things. So anything that can dissolve in water is being separated and pulled apart because of that polar, that opposite charges on the water molecule. High cushioning, chemical reactivity, solvent, and heat capacity. Okay. So that's our cool characteristics of water, and a lot of it has to do with those hydrogen bonds, the polar properties of water. All right, the next one is salts. So we just kind of got done looking at how water is a great solvent and these salts. And so here is some pictures of salts growing naturally out in these salt flats. So salts are really important because they give us what we call electrolytes. You may have heard that before, like Gatorade and Powerade and Pedialyte. These electrolytes help to maintain our biochemistry. So cells like muscle cells and neurons, they need things like sodium, potassium, calcium, chlorine. I think those are the biggies. Iron, I think, is a plus. Magnesium, I think, is a plus. So these are electrolytes, and they can create salts, but water in our bodies dissolves them. And so now we have all these positive and negatively charged electrolytes floating around in our system, but that's important for our cells to be able to work. So we're going to see some of that when we get into the muscle physiology and the neurophysiology in chapters 6 and 7. Okay. The next major topic before we get to organic molecules is acids and bases. So you may have heard this before or have heard of the pH scale. So when we talk about acids and bases, these are things that have, we're measuring the concentration of hydrogen ions. So a hydrogen ion is the H+. And all that means, if you remember, it's got one proton in the middle. And that's it. It's lost its electron, because remember, a hydrogen atom was one proton and one electron. So it's lost its electron. So all it is just a proton floating around. So sometimes you hear of hydrogen ions being called a proton, because that's really all it is. So in acids and bases, there is a indirect relationship, inverse relationship between concentrations of hydrogen and this value on the pH scale. So if you have high levels of hydrogen ions, means you have a whole bunch of these hydrogen ions floating around in your solution, that's going to give you a lower pH number, less than 7, right? So neutral is 7 right here. Okay. So if it's less than 7, we have a higher concentration of hydrogen ions. If your hydrogen levels are low, that's going to give you a higher pH number. This is going to be greater than 7. And we have words for these. So when you have something that's less than 7, it's got a low pH. This is an acid. When you have hardly any hydrogen ions, the pH number is high, greater than 7. This is what we call a base. Okay. So here we have this side is 0 to 7, or 6.9. Those are our examples of acid, like lemon juice, gastric acid, tomato juice, coffee, saliva, milk. These are all slightly acidic to really acidic. On the other side, blood, ocean water, milk, magnesia, ammonia, bleach, oven cleaner, these are all bases. They have lower concentrations of hydrogen ions. Okay. Acids, bases. I'm just looking at the notes here to make sure we understand. So some common acids, like in our gastric juice, try to put a biological spin on this, is hydrochloric acid, HCl. And HCl is basically a hydrogen ion plus a chlorine ion, ionically bonded together. This is what's in our gastric stomach acid, so pH of 2, which is pretty acidic. A common base we have is sodium hydroxide, NaOH. And it's basically a sodium ion attracted to a OH. OH is kind of what's called a functional group. It's two atoms stuck together, but it kind of functions as one group, to an OH minus. So if you take an acid and a base and mix them together, what happens? Well, this is called a neutralization reaction. So I'm going to erase these real quick and write them more in a reaction kind of form. So if you take hydrochloric acid plus sodium hydroxide, we are going to see one of those exchange reactions. We're going to see things swap partners. Okay, so remember, this was positive and this was negative. This was positive and this was negative. So if we're going to swap partners, we need a new positive negative combination on the reactants side, or the product side of our reaction. So let's say sodium is positive over here. We've got to swap it with the other negative. Hey, sodium chloride, we've seen that before. And now we have what's left over is an HOH, which we haven't really seen that before, have we? What if we did like this? Yeah, water. So typically, when you do a neutralization reaction, you have an acid and a base. Mix that together. What you'll end up with is water and a salt. The salt's going to be different depending on what your acid and base is. In this particular example, the salt just happens to be sodium chloride and water, but you could have a calcium chloride if you have a different kind of acid or base. Okay, all right, so now we're going to move away from acids and bases and do our last 4 slides, taking a look at organic molecules. Okay. There's 4 major organic molecules: carbohydrates, lipids, proteins, and nucleic acids. So this first one we're going to talk about is carbohydrates. And again, remember carbons, hydrogens, oxygens, and nitrogens. Carbohydrates are only made up of carbon, hydrogen, and oxygen. They don't have any nitrogen in them. And there's a couple different types of, or some various types of carbohydrates: simple sugars or monosaccharides, things like glucose. Would be an example. When you have 2 monosaccharides hooked together, it's called a disaccharide. So this would be like sucrose is a disaccharide. Sucrose is table sugar. It's like what you have in, baking or you put in your coffee. So glucose is a monosaccharide, disaccharide like glucose or sucrose, or you can have a whole bunch of them hooked together called a polysaccharide, and that would be starches, things like grains, bread, rice. That's where you're going to find more of these complex carbohydrates. You've probably heard that before. And then reaction-wise, we can actually go back and forth between monosaccharides, disaccharides, or polysaccharides. We have these types of reactions where if we're to take glucose plus fructose gives us sucrose, right? So 2 monosaccharides plus 2 monosaccharides will create a disaccharide. This is called a dehydration synthesis. There's that word synthesis again, which means to make. So we are making something new by the removal of water, dehydration. So that's just part of the bonding, and there's an enzyme that's going to be in there and helping that happen. But what if we want to break sucrose down? So when we eat sucrose, to get it absorbed into our system as glucose and fructose, we have to have enzymes that are going to come in and break that. And we use this reaction called hydrolysis or hydrolysis. Hydro meaning water, lysis means to break. So we'll break this bond, releasing water to take sucrose back into glucose and fructose. Okay? So those are very common reactions catalyzed by enzymes that drive either the synthesis reaction or the hydrolytic reaction. So those are carbohydrates, primarily used for energy. So we burn these in our mitochondria to give us that ATP. Okay. The next is lipids, also only made up of carbon, hydrogen, and oxygen. There's no nitrogen associated with lipids. There's a couple different types of lipids here. We have fatty acids, which are big, long chains of carbons and hydrogens, sometimes like 18 carbons long, saturated with hydrogens. When you get 3 of those hooked together, this is called a triglyceride. You may have heard that before, your triglycerides if you're getting blood work done. We have cholesterols, which are also fats, primarily this 4-ringed molecule that has some side chains on it. And then we have what's called a phospholipid, which is going to be really important. We're going to see this when we get into cell structure next time in chapter 3. So a phospholipid has 2 fatty acid chains and then a nitrogen and phosphate containing head. You know what? And I should say maybe nitrogen and phosphate, depending on which, if you're looking at phospholipids, they do have those extra atoms in there. But most lipids are carbon, hydrogen, and oxygen. Okay. All right, our next major macromolecule are proteins. So these have carbon, hydrogen, oxygen, and nitrogen, sometimes with some sulfur and phosphate in there, depending on the amino acid being used. There's 20 different amino acids. Now that I think about it, I don't think there's any phosphorus in amino acids. There's cysteine has sulfur. Right there is an example. So what are proteins? Well, proteins are big, long chains of amino acids. Okay, Corey, well, what are amino acids? Well, this is an amino acid, kind of this orange box. It has this skeleton, kind of this base skeleton of an amine group, that's the NH2, hooked to a carbon, and then that central carbon is hooked to an acid group called COOH, it's called carboxylic acid. So that whatever is in the orange box, if you'll notice, is the same on every amino acid. Okay, so that's that skeleton. But what is different is in the green box, and this is what we call the R group. So if you have an hydrogen, get green here, if you have a hydrogen as your R group, you are the amino acid glycine, and you have certain chemical characteristics. If you have these other atoms, combination of atoms, more different kinds of atoms, these are different amino acids. So this shows us glycine, aspartic acid, lysine, and cysteine. And so what happens is these are selected. The instructions for making proteins are found in your DNA, which we'll talk about more in chapter 3. And then what we get is this long chain of these amino acids in this very specific order, right? So very specific order of these amino acids, and those side chains allow them to react with each other. Sometimes they are attracted, sometimes they repel. And what we end up is this three-dimensional folding to get your final protein shape. So this is called primary, is just the sequence of amino acids. When they fold, their first level of folding is called secondary, and it can either be a pleated sheet or alpha helix. Then that secondary, those sheets or the helices fold again, and we get what's called a tertiary structure, and this is like a blob. It's just like it's a big molecule, and that's our protein. Sometimes multiple tertiary groups hook together, and this is what we call quaternary. So it's like 4 levels of protein structure and protein organization: primary, secondary, tertiary, and quaternary. Some examples of some quaternary proteins: hemoglobin. Hemoglobin is a quaternary protein with 4 tertiary blobs, right? So I'll try to track this one right here. It's folded, and it's got the iron-containing heme in the middle. That's where the oxygen binds. So there's 4 of those blobs, and that makes up your hemoglobin. Or collagen, which is in your hair and your skin and your nails, is 3 long linear protein chains. That would be a quaternary protein of 3 tertiary structures all twisted together to make that nice, strong, fibrous protein called collagen, part of your connective tissues and hair and nail and bone and stuff. All right, so those are proteins. Proteins function not so much for energy, like carbohydrates and lipids serve primarily for breakdown to release that energy. Proteins can be enzymes, which are catalysts for chemical reactions. Proteins can be structural, like we saw the collagen. They can be globular or transport proteins like hemoglobin. They can be enzymes, which trigger chemical reactions. Did I say that already? I can't remember. They can be ion channels embedded in plasma membranes. We're going to see proteins a lot throughout the rest of all of these body systems. All right, and then our last organic compound or macromolecule are nucleic acids. These are primarily going to be your DNA and your RNA. So we'll see this a little bit in chapter 3 when we take a look at cell structure and function. But this is your genetic code. It is what holds the instructions for making all of those proteins that we just got done talking about, or they serve a purpose in the replication of the cell when it makes a copy of itself, or to make those proteins. The RNA gets involved. So nucleic acids are genetic information. We don't use them for structural things. We don't use them for energy. We do not break them down for nutrients. They are just living in the nucleus of the cell, holding on to all of that genetic information. Okay. And then lastly, just a quick discussion about ATP, since we're going to see that quite a bit. ATP is related to nucleic acids. It's adenosine triphosphate. You can see these 3 phosphate groups on here. It is the bond right here between that third phosphate group and the second phosphate group, that when that is broken, it releases a lot of energy. And that's the energy for the cell to do work. So if you remember, our mighty mitochondria takes in our chemicals from the food that we eat, converts it into ATP, right? So we're going like this. When we break off that third phosphate group, we get this big release of energy, and that allows our cells to do the work that they need. And so ATP is sometimes called the energy currency of the cell. It allows the cell to do all the things it needs to do. Without ATP, our cells would not function. All right, I think that's it. So that wraps up our biochemistry, kind of these more biologically cell-related chemistry topics. All right, so I'll see you next time for cell structure and function from chapter 3. Bye.